Okay, in this video we will discuss about the various processes that takes place in the glycolysis. Okay, so far we have learned that the breakdown of glucose to pyruvate is known as glycolysis. The breakdown of glucose to pyruvate is known as glycolysis. But there are 10 steps involved in the glycolysis. 10 steps involved in the glycolysis. Okay, so soon after cellular respiration, a molecule of ATP is synthesized. A molecule of ATP is synthesized. This ATP is known as energy currency of the cell. Energy currency. This ATP is known as energy currency, which provides. So this ATP is known as energy currency, stored form of energy, right? Okay. So glycolysis, we are going to discuss about glycolysis and the 10 processes that are involved in the glycolysis. And glycolysis is a common pathway in aerobic respiration as well as in anaerobic respiration. The first step is known as glycolysis. Okay, the first step is known as glycolysis, and we know that it is a common pathway for aerobic respiration as well as anaerobic respiration. Okay, and so the first point we need to understand is it is common for both aerobic respiration and anaerobic respiration. Okay, this is the first point we need to keep in mind. And the site where this glycolysis takes place, is the cytoplasm. Okay, the site where the glycolysis takes place is the cytoplasm. Okay, the first step of aerobic and anaerobic respiration is glycolysis. So first it takes place in the cytoplasm. Okay, cytoplasm, and this glycolysis is also known as EMP pathway. Okay, EMP pathway. Why it is called EMP pathway is, okay, there were 3 scientists who discovered this pathway. So based on their names, this EMP pathway is given. Okay, so E means Emden, M means mayor of, Emden mayor of, and Parnis. So Emden, mayor of, and Parnis. These were the 3 scientists, okay, who gave this EMP pathway. The other name for glycolysis is also known as EMP pathway. Don't confuse. Okay, what is EMP pathway means glycolysis. What is glycolysis means EMP pathway. So these are the 3 points we need to keep in mind before we go further into this glycolysis process. Okay, so first we will start with glucose. Okay, first it starts with glycolysis starts with glucose, and this glucose gets converted into glucose 6-phosphate. Okay, and here ATP is invested. Okay, that means ATP is used. There are 2 phases here in this process. In one phase ATP gets used up, and the next phase ATP is given back. Okay, that means here invested and the ATP is synthesized. Okay, so here ATP is getting used up. So when glucose converts into glucose 6-phosphate, one molecule of ATP gets reduced into ADP. Now the ATP becomes ADP. Okay, and here what is added? Phosphate. Okay, here phosphate is reduced to the glucose and added what? Phosphate. That's why ATP is required to add what? Glucose as well as energy. Okay, so ATP gets converted into ADP. Next, the enzyme which is involved in this reaction is hexokinase or phosphorylase. Hexokinase or phosphorylase. Okay. Next step after glucose 6-phosphate is this glucose 6-phosphate gets converted into fructose 6-phosphate. Fructose 6-phosphate. Glucose, we know that glucose is C6H12O6. Okay, and fructose is also C6H12O6. Okay, there is no difference in the molecular formula, but there is difference in the arrangement. Okay, there is difference in the arrangement of atoms. That's why fructose and glucose, they are having similar molecular formula but different arrangement. So glucose 6-phosphate gets converted into fructose 6-phosphate. Okay, here hexose sugar. 6 means what? Hexose. So here hexose sugar is getting converted into another hexose sugar. Another hexose sugar. Okay. And the enzyme that is involved in conversion of glucose 6-phosphate is phosphoglucoisomerase. Phosphoglucoisomerase, or we can also say glucose 6-phosphate is converted into its isomer by isomer forming fructose. Isomer forming fructose. Why fructose? Okay, why here fructose? Because I told you it is just like similar molecular formula. That's why it is isomer. Glucose gets converted into its isomer known as fructose 6-phosphate. Fructose 6-phosphate. And here again the enzyme is glucose isomerase. Glucose isomerase. And next step is fructose 6-phosphate gets converted into fructose 1,6-biphosphate. 1,6-biphosphate. Okay, here again one more phosphorus is added. One more phosphorus is added. Biphosphate. So one more phosphorus is added, and for this it is ATP that provides phosphorus as well as energy. Okay, when ATP is used, it gets reduced into ADP. Okay, so ATP is what? Adenosine triphosphate. Adenosine triphosphate is the full form of ATP. Now ADP, adenosine diphosphate. Adenosine diphosphate. Okay, adenosine triphosphate, adenosine diphosphate. Next, this glucose 1,6-biphosphate gets splitted up into two molecules. They are hydroxyacetone phosphate and glyceraldehyde 3-phosphate by the enzyme known as aldolase. Okay, before reaction fructose 6-phosphate gets converted into fructose 1,6-biphosphate, and the enzyme is phosphorylase. Which converts fructose 6-phosphate into fructose 1,6-biphosphate. And fructose 1,6-biphosphates, they get splitted up into two molecules, and the enzyme is aldolase. Okay. Next, this dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, they can interchange. That means they are interchangeable. This can be converted into this, and this can be converted into this. That's why they are isomers of each other. Okay. From here we continue with glyceraldehyde 3-phosphate, and we will keep a full stop here. For example, full stop. Now glyceraldehyde 3-phosphate gets converted into 1,3-biphosphoglycerate. And this is redox reaction or oxidation reaction. This conversion is oxidation reaction. If something has to get oxidized, okay, it should lose something, right? something has to get oxidized means something should be loosed. Okay, in oxidation reaction we know that if something is oxidized, something should be again reduced. Okay, something should be again reduced. So what is that helping to oxidize and what is that it is reducing is NAD. Okay, NAD plus. NAD means nicotine amide, adenine diphosphate. Okay, dinucleotide. Okay, adenine, nicotine amide adenine dinucleotide is the NAD. Okay, NAD plus gets reduced into NADH plus H. NADH plus H plus. NADH plus H plus is the reduced form of NAD. Okay, I told you if something has to get oxidized, something should be reduced. So NAD, okay, is getting reduced into NADH plus and H plus. Okay, in this reaction, because this is redox reaction, you will be studying in chemistry. Okay, this is redox reaction. Okay, now glyceraldehyde 3-phosphate gets reduced by this enzyme and forms what? 1,3-biphosphoglycerate. 1,3-biphosphoglycerate. Okay, 1,3-biphosphoglycerate, and this is the energy currency, which the conversion of glyceraldehyde 3-phosphate, okay, is 1,3-biphosphoglycerate. 1,3-biphosphoglycerate. That means glyceraldehyde is an aldehyde which gets reduced by redox reaction and forms what? 1,3. Okay, so glyceraldehyde is an aldehyde which gets oxidized and form 1,3-biphosphoglycerate. 1,3-biphosphoglycerate, and here NAD plus gets reduced into NADH plus H plus. Okay, what is the full form of NAD? NADH plus H plus means what? Same thing you can write, nicotine amide adenine dinucleotide. Okay, but reduced form. Okay, reduced form of NAD. Reduced form of NAD is nicotine amide dinucleotide. Okay, so this is reduced form, and this can be also written as NADH2. NADH2. Okay, this can be written as NADH2. Okay, next is 1,3-biphosphoglycerate gets converted into 3-phosphoglycerate. 3-phosphoglycerate, and here again one more. Here synthesis of ATP takes place. Now this is the phase 2. Okay, and from here that is the phase 1. In phase 1, investment of ATP, and in phase 2, synthesis of ATP. Synthesis of ATP. Now, in order to put phosphate molecule in this, there should be some molecule which should add ATP. So ADP is the ADP reduced and form what? ATP. Now here ATP synthesis takes place. So 1,3-biphosphoglycerate gets converted into 3-phosphoglycerate, and ADP gets converted into ATP, and now ATP synthesis takes place. ATP synthesis takes place. Okay, now this 3-phosphoglycerate gets converted into 2-phosphoglycerate. Okay, and here the enzyme is phosphoglycerate mutase. Phosphoglycerate mutase. Okay, that means what? The phosphorus or the phosphate molecule which is in 3 carbon gets shifted into 2 carbon. That's why mutase. Okay, phosphoglycerate mutase. Next, this 3-phosphoglycerate gets converted into 2-phosphoglycerate. Okay, and that enzyme is phosphoglycerate mutase. Now this 2-phosphoglycerate gets converted into phosphoenolpyruvate, another acid, phosphoenolpyruvate, and the enzyme that is involved in conversion is enolase. Enolase is the enzyme. Enolase is the enzyme. Okay, enolase is the enzyme. So next, this phosphoenolpyruvate is finally converted into pyruvic acid. Finally converted into pyruvic acid. Okay, and here again the ADP gets utilized and form what? ATP. So ATP synthesis takes place. Okay, ATP synthesis takes place. Pyruvic acid do not have any phosphate molecule, whereas phosphoenolpyruvate, it is having one phosphate molecule. That's why it gets reduced into ATP. Okay, ATP is known as adenosine triphosphate. Okay, so here again one more ATP synthesis takes place. ATP synthesis takes place. So far 2 ATP synthesis took place, and here 2 ATP we utilized here. 2 ATP is used, 2 ATP is synthesized. Okay, and here I have told you dihydroxyacetone phosphate can get converted into glyceraldehyde 3-phosphate. So this is interchangeable. Once this glyceraldehyde 3-phosphate gets further reactions, and finally, okay, once its task is completed, this dihydroxyacetone phosphate gets converted into glyceraldehyde 3-phosphate, and again further reaction takes place. That means we have used how many ATP? How many ATP synthesis took place? Here 2 ATP. 2 ATP synthesized, right? 2 ATP synthesized. This is very important. Okay, 2 ATP synthesis took place. So first with this 2 ATP, and next this will get converted into this and gets again 2 ATP. That means 2 plus 2 ATP equals to 4 ATPs. Okay, we got 4 ATPs, and here 2 ATP we have used. 2 ATP we have used. Okay, this is utilization process and this is synthesis process, right? So 1 and 2. So 2 ATP we have used. So we have to minus 2 ATP. That means how many ATP we will get at? Okay, 2 ATP is the final ATP. That means net amount of ATP is 2 ATP. Okay, now, and we need to remember that NADH plus H plus has the tendency to produce 3 ATP. Has the tendency to produce 3 ATP. Okay, so here first reaction 1 NADH. Okay, let me take it as NADH2. Okay, in first reaction NADH2 1. In second reaction again it gets converted and again goes for the further reaction, again one more NADH2. That means how many? 2 NADH. 2 NADH2. Okay, they can have the ability to produce 3 ATP. Once 3 ATP, and the next time it gets converted again 3 ATP. That means total 6 ATP. Total 6 ATP. Okay, 6 ATP is produced here. Okay, so we from 2 NADH2 we get 6 ATPs, right? Okay, once in this reaction 1 NADH2 and another reaction which gets converted and goes for the further reaction, we get 2 NADH2 and we get 2 NADH2 means 3 ATP, each will produce 6 ATPs. 6 ATPs. Now what we need to keep in mind is, in this reaction we get total net amount of ATP is 2 ATP, and if we see total gross amount of ATP, gross amount of ATP is 8 ATPs. Okay, 2 plus 6 ATP is 8 ATPs, and these 6 ATPs, okay, which are formed from NADH2 are not used immediately. They can be formed as check. Okay, they can be formed as check, and they have to be converted into currency, and this is the immediate currency that we can use. So 2 ATP is the acts like immediate currency, and this 6 acts like a check, okay, which gets further conversion and forms what? Currency. So total gross is 8. Okay, 6 plus 2 is 8, and we know that from when in this glycolysis process glucose gets converted into pyruvic acid, first with this one pyruvic acid, and finally this gets converted and another pyruvic acid. So how many pyruvic acid in this reaction? There will be 2 pyruvic acids. Okay, so we need to keep in mind this 2 ATP net amount. Okay, so the gross amount of ATPs are 8 ATPs. Now we need to keep in mind that in this glycolysis process 2 pyruvic acid is formed and 2 net amount of ATP, and the gross amount of ATP is 8. Okay, 8. So all these things, they are having 10 steps. Okay, in glycolysis 10 steps. First one is glucose gets converted into glucose 6-phosphate. Okay, then the third one is fructose. This glucose 6-phosphate gets converted into fructose 6-phosphate. Third one. And this is the fourth one, fructose 1,6-biphosphate, and this is the fifth one, and 6, 7, 8, 9 and 10. Okay, so there are